Okay, thank you. It's kind of weird sitting here because there are 3 or 4 people around me managing screens and so forth. I can't see you, but I'm looking in this monitor. I'm going to pretend that you're all out there in front of my classroom. So this is a session on CRISPR, and it used to be that I would start off by saying that CRISPR is the hottest topic in biology right now. I can't say that any longer because, of course, this is now the hottest topic in biology, and I guess no one can really see me. I'm holding up a model of the coronavirus. So for the last 6 months, CRISPR has kind of faded to the background while people have focused on coronavirus, and we're actually doing a workshop tomorrow that's focused on coronavirus. So I won't say anything more about that right now. We're going to go back to CRISPR. CRISPR, especially this protein right here, this is known as the CRISPR Cas9 protein. This is what we're going to begin to use to edit the human genome. So that's what this next 40-minute session is going to be all about. Jennifer Doudna's photo was up there, so I should start by acknowledging the recent Nobel Prize in Chemistry. That's Jennifer Doudna at UC Berkeley. And you'll notice she has a very interesting physical model of the CRISPR Cas9 protein in her hand. That was designed for Jennifer's lab, and we'll be going over that model today. So there's going to actually be 3 parts to this 40-minute presentation. In the first part, we're going to talk about how are you going to connect this topic of CRISPR to something you already teach. And to do that, we're going to be comparing the CRISPR Cas9 endonuclease with a restriction enzyme. Okay, and then so that's the first part. Second part, we will talk about CRISPR biology. That is, how does this CRISPR system function as an adaptive immune system in bacteria? And then the final part, we will look at the Cas9 endonuclease in more detail. And we'll go through a very schematic foam-based model of CRISPR-Cas9. Okay, to begin then, how are you gonna bring up the topic of CRISPR to your students? So what I'd like to suggest, there are many ways you can do that. This is only one way that I'm going to suggest, and that is your students, depending upon if they're freshmen or in some upper level course, they may already have heard about restriction enzymes. So I have here the EcoR1 restriction enzyme. So this is a nuclease that can bind to a specific sequence of DNA, and it can cut it at a very specific sequence. These restriction enzymes were discovered in the early 70s, and they resulted in the recombinant DNA era. We started being able to analyze genes in small genomes like viruses or plasmids. We were able to clone DNA fragments. We were able to sequence them. We're really able to start to do what we now call molecular biology. So it's an endonuclease that recognizes a specific sequence of DNA and it cuts it. Why didn't we begin using restriction enzymes to edit the human genome way back in the 70s? Well, we didn't start using them because they weren't up to the task. What we need in order to edit the human genome is we need an endonuclease that can cut at a statistically unique site in the human genome. The human genome is 3.2 billion base pairs in length. And this enzyme can seek out and find an absolutely unique sequence in that 3.2 billion base pairs and make a cut. So if we wanted to edit the human beta globin gene, we could program this CRISPR-Cas9 to seek out and cut the human beta globin gene only once at one specific site in that gene. And that's the first step then to being able to edit that gene. So you all received, I think, a student modeling pack that had some nucleotides in it. And if you've done your homework, you may have already this 16 nucleotide or 16 base pair long sequence in front of you. So if you do, bring that out, and we're going to do just a quick activity here. I'm going to suggest a way that you can get your kids to think about the difference between a restriction enzyme and the CRISPR-Cas9 endonuclease. So if I can have the overhead camera on, Heather, we'll look at this sequence here. And I don't know if any of you looked at this sequence very carefully, but we've hidden an EcoR1 restriction enzyme site right here in the middle. I'm actually going to just pop it out of there. So these 6 bases here, this GAATTC, that's the recognition site for the EcoR1 restriction enzyme. And of course, it's a palindrome. So this same sequence, 5' to 3' on this strand, is shown again on the opposite strand, 5' to 3', oops, 3' GAATTC. So that's the interesting feature of restriction enzymes. They recognize palindromic sequences of somewhere between 4 to 6 base pairs long, and they cut the DNA. Let me just show you one quick thing. We'll put this back in a long sequence. When the EcoR1 binds here, it cuts one strand on this side, and then it cuts the other strand on at the symmetrical side on the other strand. And as easily as that, this restriction enzyme has cut this DNA. And in this case, you have these so-called staggered ends, sticky ends, that prove to be so useful in cloning DNA. Okay, so here's what I'm going to suggest that you do. The activity that I'm about to briefly describe to you in about 10 minutes here is actually described in more detail in a handout that you'll find available at the end of this session. So follow along as best you can here, but know that there's a written description of this activity that will follow. So what I'm going to suggest is that you give your students a bunch of these foam nucleotides. And then what we're going to ask is, how often would we expect to find this EcoR1 restriction site, this GAATTC, in a genome, okay? And let's assume we're talking about a genome that's just a random sequence of nucleotides. All right, how often would you expect to find an EcoR1 cleavage site? And what you might say to your students is, what if you were just to start putting these nucleotides together in any random sequence? How many of these sequences would you have to put together before you would have accidentally, just by random, created a GAATTC? All right, so you start the activity like this. Say, put these nucleotide pile in front of you, just reach in with your eyes closed, pick out one, put it down in front of you, and I just happened to get a G. What's the chance that I would have picked out a G, the first letter in this sequence that we want to make? Well, the chance is 1 of 4, because there are 3 other kinds of nucleotides I could have chosen. So 1 in 4, okay? Then close your eyes again and reach in, choose another one, and I happen to get an A. So GA. What's the chance that I would start by making a GA? Well, that's 4 times 4, that's 16. So if you have a class full of 16 students, let's say your class is 32 students, and if they all made 2 nucleotides at random, you would expect 2 of them, 1 out of 16, to have a GA. Okay, that's an experiment you can do with your students in your class. All right, close your eyes again, pick out another nucleotide, and of course I'm cheating, right, because I'm looking. I picked out another A, so we get a GAA. What's the chance that anyone would have made a GAA? And that's 4 times 4 times 4, 16 times 4, that's 64. So another way to think about this, have all of your students make a 64 nucleotide long random sequence. And then they've all got their sequence, and then you ask them to scan down that sequence of random nucleotides with some sort of with this sequence that you want them to find, and ask them if any of them made, if any of them made a GAA, okay? And the prediction here in this experiment we're doing is that if they made 64 nucleotides of random sequence, one of them would all of them should find one example of a GAA trinucleotide sequence. All right. I'm going to run out of time here quickly, so I'm going to move along. We keep multiplying 4 every time we add another nucleotide, so that by the time you get a 6 nucleotide sequence here, you've multiplied 4 by 4 by 4 by 4 by 4 by 4. The answer is 4,096. So we would expect to find an EcoR1 restriction site every 4,096 base pairs. Let's say every 4,000 base pairs in random DNA. Why can't we use EcoR1 then, or other restriction enzymes, to start editing the human genome? Human genome is 3.2 billion base pairs long. If you divide 3.2 billion by 4,000, what you would find is that EcoR1 will cleave the human genome 800,000 times to make 800,000 different fragments, all of different sequences. They would all end with an EcoR1 restriction site, but the sequences in between would be just random sequences. So restriction enzymes, no one even thought of editing the human genome with restriction enzymes. They're just not up to the task. So what's most amazing then is that starting 30 years ago, people began to discover CRISPR. And I wish we had time to really talk about how that happened. We don't have time to talk about the history, but it's a fascinating story of science. So this enzyme was in those same bacteria all that time, all through the 70s, the 80s, 90s, when we were using these to clone and sequence DNA. We eventually discovered this enzyme. We eventually figured out how it was working. So by about 2012, there was a classic paper published that really showed that this CRISPR system functioned as an adaptive immune system in bacteria. And that's when the whole CRISPR field kind of blew wide open. And just recently then, Jennifer Doudna and her colleagues received a Nobel Prize for all of this early work. So the question then is, what I want to tell you is this enzyme, if you add it to the human genome, it will cut that genome once. And we're going to program this endonuclease with a piece of orange guide RNA. And that guide RNA is going to direct this endonuclease, Cas9, to a specific sequence of double-stranded DNA. That's the key difference between Cas9 and a restriction enzyme. A restriction enzyme just binds DNA and cuts it. Cas9 is equipped with a guide RNA that base pairs with a specific sequence of target DNA, and only if it's a perfect match between its guide RNA and a strand of the double-stranded DNA, target DNA, only then, if it's a match, will it cut the DNA. That's the big difference. In the last part of this session, we'll look more carefully at that. But let's go back and do some calculations. So we know that any 6-base sequence, then, any 6-base pair sequence occurs once every 4,000 times. How many more nucleotides would we have to add onto the 6 base pairs in order to be looking at a statistically unique sequence, a sequence that we would predict would only occur once in the human genome? And don't tell your students the answer to this question. I'm going to give you the answer right now. So look away if you don't want to know the answer. The answer is your homework. This 16 base pair fragment, this represents a statistically unique sequence in the human genome. But have your students do the following calculation. Take them back to the EcoR1 restriction site that occurs once every 4,000 base pairs, and have them start adding additional nucleotides. What's 4,000 times 4? It's about 16,000. Add another one. What's 16,000 times 4? I don't know. That's getting too big for me to do in my head. Point is, you keep adding a nucleotide and multiplying by 4 until you reach a size, a number, that is equal to or greater than 3.2 billion nucleotides. And if you do that, you will find, you will arrive at this answer, a 16 base pair piece of double-stranded DNA would statistically be predicted only once in the human genome. So that's what's special about the CRISPR system. We have here a new toolbox that we can add, a new tool we can add to our molecular biology toolbox, and it allows us to do something that we've never been able to do before. And that is to deal with these incredibly huge eukaryotic genomes. And now we start making specific cuts in those genomes with this Cas9 enzyme. All right, so.
We're right on time.
All right, that was perfect timing. Yeah, I'm amazed. I don't know if that made any sense, but again, this activity is all written down in a handout that you'll find at the end of the session. So we're going to move on right away then. And when we started...
You're supposed to ask for some questions.
Are there...
Give people some time.
Yeah. Are there any questions anyone would like to ask? You can unmute yourself and shout it out, or you can type it in the chat. I'm not able to actually see the chat, but someone is monitoring the chat. If someone wants to ask a question, I would be happy to respond to any questions you might have. Learning is a slow, gradual, transformative process shaped by conversation. So there'll be very little learning that goes on in this session because we're going to rush. And it's really hard to have conversations.
Do you have a question? And is there anything else that needs to be present for the Cas9 to work?
The answer is no. That's what's beautiful about this system. The only thing is this guide RNA. So it's not just the Cas9 protein. It's actually a protein RNA complex. So as long as you have equipped this Cas9 protein with a guide RNA to tell it where it should go, what it should search for, it will do that and it'll make the cut. That's what makes it such a useful tool. Okay, any other questions?
While you guys are thinking about that, Chris has the screen showing some of our free digital materials that you can actually drag and drop different labels, and we have all of these that are associated with our student modeling packs.
Right. So these, of course, if you've been to other sessions, you know, these modeling packs were created specifically for your students to use in these virtual environments from home. So
we are getting a couple of other questions. And so one is, how many base pairs does the guide RNA have? Oh, not base pairs, but nucleotides.
Nucleotides, yeah. Mm-hmm. Well, in the last section here, you're actually going to see that. And the answer, I don't know the answer, but it's on the order of maybe 50 to 60 nucleotides long. It's actually initially made up of 2 RNAs, which Jennifer Dowd had turned it into a single guide RNA, and it won her the Nobel Prize.
So, and I had thought that you had said before that the part that actually finds the correct sequence is about 21 base pairs.
Yeah, the business end of the guide RNA is about 20 nucleotides long. But there's a lot of other RNA that is actually recognized by the Cas1 to bind the guide RNA such that the 20 base pair end, the 20 nucleotide end, is positioned just right to find the target DNA strand.
Okay, we should be moving on to our next video.
Our next video, then, we're gonna play a video here that describes how CRISPR-Cas9 works. How the CRISPR system works as an adaptive immunity system in bacteria. Bacteria protect themselves from bacteriophages, with CRISPR systems. So we'll play this video, and then we'll come back and see if it generated any questions. So one of the things we realized when we started talking with teachers about CRISPR, we were very anxious to just talk with them about how this Cas9 enzyme is used as a tool by molecular biologists to edit a human gene. But of course, we learned very, very soon that teachers are really interested in viruses, and they want to be able to talk to their kids about what is a virus. Even before you get to coronavirus, they want to talk about viruses in general. So, when we just casually mentioned that CRISPR is an adaptive immunity system in bacteria, it was that protects the bacteria against viruses, against bacterial viruses known as bacteriophages. Then that really caught the teacher's interest. So we backed up at that point then and started thinking about how do you teach CRISPR biology. So here's what we call the CRISPR Adaptive Immunity Kit that you could use to explain to your students how this system works in bacteria. Up here we have 3 bacteriophages. And we know they're different because you can see that the double-stranded DNA genome contained by each one of these viruses is colored a different color. And this is the bacterial cell membrane. So these guys are on the outside, and they're about to try to infect the cell by introducing this double-stranded viral DNA into this cell. So this virus here with the purple genome is settling down on this membrane, and in a complicated molecular process, it's going to inject its DNA into this cell. Now, we can see here that, in fact, this bacterial cell has had a previous run-in with this particular purple virus. And we know that because in a particular section, a small section of this bacterial genome, there's something that's called the CRISPR array. And it's a sequence of genes that people over the last 30 years have come to recognize as being diagnostic of, a CRISPR system. This bacteria has a CRISPR system. Not all bacteria have CRISPR systems, only about 50% of them. So that's another side question that we could talk about if we had more time. But this bacteria has survived a previous attempt by this purple virus to infect it and kill it. And it survived because it was able to capture a small fragment of this genome and incorporate it into this CRISPR array. And then from this CRISPR array, deploy a series of Cas9 proteins programmed with some RNA that makes it possible for this Cas9 to provide constant surveillance for the entry of this purple bacteriophage genome into the bacteria again. And I just said a lot in the last 30 seconds. So let me go back and try to go through that more slowly then. So as an adaptive immune system, we're going to adapt to a prior exposure to the purple virus by deploying a bunch of enzymes that we're not dealing with here in this very schematic, simplified view of CRISPR. But we're going to send some enzymes out to cut up this viral genome, this purple genome, into pieces. And then we're going to bring one piece back and we're going to integrate it into this CRISPR array in the bacterial genome, just between these 2 black diamonds here. And let me stop right now and let's look at this CRISPR array. This is what that Japanese group discovered and reported in the sequence that was just downstream from an E. coli gene that they were sequencing. What they reported were some short palindromic repeats. See right here, short palindromic repeats. They noticed these sequences, which were about 30 nucleotides long, and they were clustered together. That's where the clustered comes from. But then they were regularly interspersed with these different colored sequences. The different colored sequences were also the same length, about 29 to 30 nucleotides long. So they were regularly interspersed with the same size of a sequence, but these were different sequences. These short palindromic repeats were almost identical, which is what called their attention to this unusual sequence in the first place. All right, what we know now, after after many, many different people looking at this and having ideas as to what this might represent, what people finally recognized is that, the sequence that is interspersed between the short palindromic repeats, these are viral sequences or sequences from other pathogens that are trying to infect this bacteria. So that was really the whole key then, that, oh, this might be an adaptive immune system, that you express or you transcribe the Cas9 gene, which is this white gene. You make a piece of messenger RNA, which is then translated by a ribosome to give you these 3 proteins, Cas9 proteins. Okay? Now, notice that this gene for Cas9 was transcribed into messenger RNA and translated into protein. These other genes were also transcribed by an RNA polymerase into RNA, but these are not messenger RNAs, these are structural RNAs. And what I've just said is that the tracer RNA is the name we give to this orange piece. The tracer RNA is complementary in sequence to a portion of the short palindromic repeat, and then that dual RNA is bound by a Cas9 protein. And the key here is that this RNA is going to program this particular Cas9 to look for a purple bacteriophage DNA sequence, while at the same time, you're going to form this dual guide RNA that's going to associate with another Cas9 enzyme. And this one is programmed to now search for a yellow bacteriophage genome, and similarly. So, depending upon this bacteria's previous exposure to viruses, it's going to deploy a whole series of RNA-programmed Cas9 enzymes that will constantly surveil the inside of this bacteriophage on the lookout for different, I didn't mean bacteriophage, constant surveillance of the bacteria for the introduction of bacteriophage sequences. And when these Cas9 enzymes find that particular sequence, it's going to cut it and inactivate that bacteriophage genome. So that's the adaptive immunity system.
Okay.
All right, we're going to go on to the next and unfortunately the last section then, and it has to do with, let's look at the nitty-gritty of the Cas9 protein. I wish each one of you had one of these in your hands, because then we could talk in great detail about how this guide RNA is bound by the Cas9 protein, and then how it base pairs with with one strand, the target strand of double-stranded DNA.
Tim, I'd like to jump in here and ask you to remove the base.
Remove what?
The base off the model. Thank you.
How's that? Okay. All right, here's the double-stranded DNA that comes in. I'm not sure if I have enough time to do this before we go to the video.
You're okay.
Double-stranded DNA comes in here, light blue strand, dark blue strand. As soon as it binds, it separates those 2 strands of DNA. And then the orange guide RNA interrogates the dark blue target strand to see if it's complementary and Watson-Crick base pairs with the guide RNA. If it is, it activates 2 different nuclease active sites in this nuclease. Each one cuts a different strand. The net result is that you introduce a double-stranded break into the DNA. And again, that is at one time all that Cas9 does, but we really have to emphasize what an amazing thing that is, that you can look through 3.2 billion base pairs of sequence and only cut once at a sequence which is complementary to about 20 nucleotides of this guide RNA. That's truly remarkable. Let's go right on to the last little video. It's an 8-minute video, and in that we try to explore this Cas9 protein using a schematic foam-based model. And we have now here on the table a large format, still schematic, but an enlarged model of that Cas9 protein. And what you see here is that the white foam on this placemat, this is the protein. It's a large protein. I think it's about 1,500 amino acids long. It has multiple domains on it. Here's the guide RNA. But before we go through this in detail, let me show you this model. So this is a 3D printed model based on the atomic coordinates of the actual structure of this protein solved by X-ray diffraction. And I'm going to just remove one of the many domains of protein structure from this 3D printed model so that you can better see how it binds to DNA as well as and how the guide RNA interrogates the DNA. So we have a double-stranded DNA that's made up of a light blue and a dark blue strand. And the double-stranded DNA comes into the Cas9 enzyme here. And you won't be able to see this, but there's a particular sequence in this double-stranded DNA that's referred to as the PAM sequence. PAM stands for protospacer adjacent motif. For Cas9, it's NGG, which the N means it can be anything, any of the 4 nucleotides followed by a G and another G. So whenever this enzyme finds 2 Gs, it'll bind to that double-stranded DNA. And then the first thing is it does, it separates the light blue from the dark blue strand. And it has bound this orange guide RNA in such a way that the end of the guide RNA then attempts to base pair with the target strand, the dark blue strand of DNA here. And only if it can form perfect Watson-Crick base pairs with that dark blue strand over a 20 nucleotide long sequence will this enzyme become active. And when I say active, there's an enzyme, there's a nuclease active site here, which will cut the dark blue strand, and there's another enzyme nuclease site over here that will cut the light blue strand. And let me see if I can do this then. So there's the double-stranded cut that this enzyme can make only if the guide RNA is exactly complementary to the dark blue DNA strand. So that's what the Cas9 enzyme does. That's how it works in its real 3-dimensional structure. So again, the white is the schematic Cas9 protein. And then the guide RNA, just as in the previous adaptive immunity kit, it's represented by 3 different colors of foam. In this model, all of the RNA is orange. And I'll just point out again that RNA folds up into a complex 3D shape just like protein. So keep that in mind. This white Cas9 protein binds guide RNA in a very specific way so that 5' end of the guide RNA is poised, it's positioned just in the right spot where it can interrogate this dark blue strand of target DNA. All right, so while it's all orange here, here's the orange part that was transcribed from the tracer gene. And then here's the black part that was transcribed from the short palindromic repeat. And then here's the purple spacer that was in between the 2 palindromic repeats. And then this thing on the end here, this is what's going to win Jennifer Doudna a Nobel Prize, because she and her colleagues figured out that they could actually connect these dual RNAs into a single guide RNA with this little tetraloop. So at that point, it became much easier to make these guide RNAs that would then program this particular Cas9 enzyme to recognize a DNA sequence that was complementary to this purple sequence. So this model is, of course, all glued down on this tabletop so that we can use it in presentations like this. But what your students would do is they have to construct this out of the pieces. And if they can position this double-stranded bacteriophage sequence in the Cas9 with the PAM sequence right here, the NGG, which is indicated on this placemat that you can't see. But if they position the DNA correctly, they can then ask if this top strand, the target strand, is complementary to the purple guide RNA. And if it is, and only if it is, then these two distinct nuclease sites on the enzyme are activated. So this nuclease site cuts the top strand, this nuclease site cuts the bottom strand, at which point you can remove this double-stranded fragment. And we could go ahead and take the other one apart as well. So we have introduced a cut, a double-stranded cut, in the human genome at a sequence which is statistically unique. So if you want to edit the human genome, you simply make a guide RNA in which these 16 to 20 nucleotides represent now the beta globin gene. And then you can target this enzyme to that particular gene and make that cut. Now, what does that do? Well, that only cuts the human genome in a specific gene at a specific site, which is no small feat. But that's only the beginning of editing the DNA. You then have to somehow affect a change at that particular sequence. And what happens next then is something that is too complex for us to get into in this kind of a short video session. But suffice it to say that CRISPR people are very, very clever researchers, and you'll be amazed as you learn about how different modifications of this cleavage reaction have been used then to affect more and more specific changes to the sequence of that particular gene. I'll just mention there's something called base editing now, and you start off by inactivating one of the two nuclease sites, and then you change one of these bases, actually in the non-target strand. You change it without ever cutting a phosphodiester bond. You basically deaminate a C, cytosine, to become a uridine, which then becomes a thymine. So you can change a C to a T in a non-target strand without ever cutting this DNA strand. That's amazing technology. Okay, I think we're back live. We're near the end of the session, but we have about 5 minutes here for questions. I know that we covered a lot of stuff in that last 8-minute video. I frankly don't know. I've told this story a number of times now, so it's perfectly clear to me, but that's only after having worked on this stuff for about 3 years straight now. So I've sort of lost touch with what it's like to hear about this for the very first time. I'd be happy to answer any questions you may have.
And if you don't have questions, do you have any comments about being able to use this with your classes, with your students? Are you interested in getting some materials like this if we were to release them as some products?
Yeah, I guess these two things, the adaptive immunity kit and this foam Cas9.
Go ahead, Erin. You can unmute yourself.
Okay, so I teach 6th grade, so I don't go into this much depth with CRISPR. we do the light start of traits and reproduction and stuff like that, but I mainly focus on like the Punnett square and things like that. We bring CRISPR in as more of like, because this generation they're going to be talking about designer babies and stuff like that. What other types of questions would you say could go along with CRISPR with like basic starting of genetics, I guess?
Yeah. Well, what I would do, I wouldn't necessarily use anything you've seen today in this session, with the exception of these foam nucleotides. I think these could be effectively used with kids that young, because kids like codes, right? Can we move this out of the
Yeah.
way for just a second? Kids like codes. So I think with the really young kids, these can be used to get them to understand that in our DNA is just a code.
I do have some questions.
And yeah, this is actually covered in some of our other workshops that we're doing here at this meeting and other meetings. So I don't want to spend a lot of time going into this, but this is what I would use with sixth graders. It's
Okay,
not much of an answer to your question, but maybe we can talk about that in more detail later.
so some of our questions were, how can, oh, I've lost it. So how can you insert a gene?
Yeah,
Let's do that. And then there's another one about what happens in the bacteria. So I'm
okay.
going to give this back to you, because you can use this for,
Well, this won't help, but again, these are the questions that you teachers always ask, and it's always hard to answer this. Cas9 makes the cut, and then you sort of stand back and you hope for the best, because your cells are now going to try to repair that cut. So we have not one but two different DNA repair systems that will try to fix that blunt cut. The first one is called an error-prone, non-homologous DNA repair system. And it'll just stick those ends back together again at all costs, and probably end up inserting or deleting a few nucleotides in the process. So if you're looking to knock out a gene, then CRISPR is a great way to do that. Because chances are, as you repair the cut that Cas9 made, then you will throw off the reading frame. So whatever protein was being expressed by that gene will no longer be made.
and so I thought, can I
Yeah.
ask a question here? I thought instead of making a blunt end cut, if you were to use a different Cas and make a sticky end cut and then insert new DNA,
Yeah.
then it would recombine
Yeah.
with the right gene.
All right. So you're jumping way ahead, but Cas9 is the nuclease that everybody talks about, but it's just one member of a whole family of Cas-like proteins, some of which make staggered cuts. So that's what Heather was alluding to. But if you're going to insert a corrective gene into that cut site, then it's more complicated, because first of all, you have to provide a homologous copy of the new gene you want to insert. That's not trivial. But that's what people do. They provide the corrective gene, the homologous gene, and then you still rely on the internal workings of that cell to use its homologous DNA repair system to insert that new gene into the cut site. And that's when I say you just kind of hope for the best, because you've got two competing repair systems, and clever people, as CRISPR researchers are, have figured out a way to suppress the error-prone non-homologous repair system if you're trying to insert a corrective gene. So they've been able to increase the efficiency with which you can introduce a new gene. But it's not easy. It's possible, okay, but it's not easy. And I don't think that will, 20, 30 years from now, when we are routinely editing genes, that's probably not the way we're going to do it. The way we're going to do it.
We're over time.
All right. So are we out of time, or do I
So
have
no,
1 minute?
we're over time right now, but we're just going to do a couple of wrap up things. If you want 30 seconds to wrap
Yeah.
up a thought, go ahead.
Well, all right. So all I would say is you need to come and spend 5 days with us in a workshop entitled Science and Ethics of Genome Editing. Now, unfortunately, that's an advanced workshop, and we are going to run that workshop this summer, but in fact, cohort 3 was signed up to do it last summer. They didn't get to do it because we had to cancel all the workshops. So there will be a cohort 4 It's an advanced workshop, which means that before you can come to the genome editing workshop, you have to have done another workshop, Modeling the Molecular World. So what you should do this year, this summer, spring of 2021, sign up for our Modeling the Molecular World workshop. That will then pre-qualify you to apply to the genome editing workshop in the summer of 2022, when we are certain to be back hosting face-to-face real workshops.
Okay, thank you, everyone. I'm going to end this meeting in just a minute. We have another session coming up, a live demo with protein folding and unfolding the mystery of proteins. So if you can join us for that. We didn't get to answer all the questions, but we will try to follow up via email, and you are welcome to reach out to us, and we are happy to answer those questions. So thank you for joining us today.